We're starting this afternoon with a wonderful keynote by a friend of mine, Manisha Desai. She's a professor in the School of Medicine and Biomedical Data Science, and she talks about AI, data science, and health. Manisha.
Wow. It's so good to be here with all of you. So I'm going to start by telling you why this topic is so meaningful to me. I need my clicker. So I'm a biostatistician, and when I was a little girl, I didn't know what a biostatistician was. Didn't really see myself as a scientist. I was actually interested in medicine. Medicine kind of intrigued me, but I was also very musical. I played the flute, and just saw myself as more of like a humanities type person. Didn't really think I was going to grow up and be a scientist. And in my last year of high school, my aunt was diagnosed with colon cancer. Or they weren't sure if it was colon cancer or if it was ovarian cancer, because it had spread all over. She had metastatic disease. And it was really devastating. So my mom immediately went over to England to take care of her. That's where she lived. And within just a few months of her diagnosis, she passed away. So there was no treatment for her, no surgery, no chemotherapy was offered. This was back in the 80s. And we were all really devastated by that. And that experience sort of reignited my interest in disease and health. It also raised a lot of questions for me. What if we had been able to detect the disease earlier? Would we have been able to save her? Is it possible to cure metastatic disease? Could we have bought more time with some kind of treatment? And then the other thing that was on my mind was, what if she had been here in the States? Would that have made a difference? So shortly after this, I went off to college, and I was still wrestling with that idea of like, should I go all in with my music? And at the same time, right at that time, for the first time, I discovered I wasn't terrible at math. In fact, I liked it. And I liked that I could apply it to study disease. So this became sort of my pathway to combine these two interests of mine. And it's been a really wonderful journey, because I think it's wonderful that we can apply data science to tackle some really tough public health questions, because we can answer questions with rigor. So by carefully designing studies, by thoughtfully analyzing data, we can tackle some of these really tough public health challenges. I mean, just think about the AIDS crisis, where in the late 80s and 90s, we saw this steep rise in deaths due to AIDS. And it wasn't until the onset of antiretroviral therapies that came in the late 90s, early 2000s worldwide, where we finally started to see a decrease, and the death rate started plummeting. And this was, of course, due to our wonderful retrovirologists and others working hard on this, but it was also due to the wonderful work by data scientists who were leading clinical trials to assess the efficacy of these therapies. And here we are today. We've got some wonderful AI tools. Now, of course, AI is not new, but we are sort of looking at newer applications of AI, new ways of integrating it into health. And there is so much hype around what AI can do and excitement. We're seeing AI as medicine's biggest moment since antibiotics. The revolution is coming. It's transforming healthcare. It's going to change our future. And with all of this promises comes some challenges. We're seeing, this is an article that was published last year in the New York Times, talking about the challenges we've had in vetting some of these AI algorithms. And we've seen some of this in this article, they talk about some algorithms that are giving off sort of false alarms incorrectly, some algorithms that are actually exacerbating some of these health disparities that we've been working so hard to bridge. And a lot of this has to do with our process of vetting these algorithms. So while drugs and devices, we have sort of a process for vetting them through the clinical trial framework, for example, to get approval from the FDA. This is kind of like a new landscape for us to how we go about vetting some of these AI-based algorithms. This is new terrain for us. And I believe it's really important that we evaluate these algorithms in a clinical trial setting. And actually, this is sort of what my team does. My team and I, we spend a lot of effort on clinical trials. Because, I'll talk a little bit more about the value of the clinical trial setting, but this is where we spend a lot of our time focusing. And what we've noticed is that we've had to really shift how we think about the clinical trial setting for these AI-based algorithms. And that's what I'm going to focus my talk on today. So one of the reasons that we love clinical trials, and we think of clinical trials as sort of the gold standard for assessing efficacy of an intervention, is because it has this wonderful quality of internal validity, which means that we can minimize the bias of our treatment effect for our clinical trial population. And that's a wonderful quality. What we strive for and where we struggle is with external validity, being able to address a relevant clinical question with generalizable findings. We want to make sure that our findings are relevant for the typical patient. And so I'm going to illustrate some of these ideas with this trial that I was involved with for an AI imaging tool. So the idea is that we have these AI imaging tools. They can be used to assist endoscopists to see those precancerous lesions so that they can remove them before they turn into cancer. So our goal with this trial was to assess the impact of the study. We were so excited to do the study because there had been a number of clinical trials, and here I'm listing all of the different clinical trials that had been done. So there had been some work done in these clinical trials showing a lot of the promise for these AI imaging tools. And I'm showing you the treatment effects for these that show that there's a lot of promise for this AI imaging tool. So we were very excited to run this trial in a real world setting. And here's what we found. We found that it didn't work. So when we rolled it out in the clinic, it didn't work. And that was really surprising to us. We thought what we had hypothesized was that if we put this into our real world clinic, the sites where we had access to the AI imaging tool, we would have an uptick in the adenoma detection rate. So that's how we gauge the properties of the screening tools. And what actually ended up happening was that we did no better than when we didn't have the tool. So we didn't do better with respect to adenoma detection rate. We didn't do better with adenoma per colonoscopy. So what happened? Why didn't our findings replicate? Well, we designed a pragmatic real-world trial. So we took, for example, the other trials were selective about which colonoscopies that were eligible for study. We took all comers. So even those where there wasn't a great preparation, where you didn't drink all, how you're supposed to do all that drinking. For those of you who are my age and have gone through colonoscopy, know you have to drink all that terrible stuff. So even the colonoscopies where there wasn't a good prep, we included those as well. But the crux was that in our case, the endoscopists were not aware of our hypotheses. They weren't aware of the study design. And in the previous trials, we believed that the estimates were overly optimistic for that reason. And I will say that other studies that came around this time that were conducted that were similarly pragmatic also found findings that were very similar to what we found. So this begs the question, should we give up on AI imaging tools for colonoscopy? And I'm going to say, no, we shouldn't. We actually remain optimistic about these tools that clearly identify polyps. But there's an important part of the equation, which is this human-AI synergy that's taking place. And so we have to ask this question, how, if at all, did the tool affect the behavior of the endoscopist? Did the AI imaging tool, maybe it worked really well. And so maybe there were some really tricky areas of the colon where it did highlight areas that we needed to be wary of, but maybe with counterbalancing factors. Maybe there were some areas where the endoscopist saw something, but it wasn't highlighted by the tool, so the endoscopist said, okay, forget about it. So maybe it yielded a lower quality exam overall because of a false sense of security. So we have to ask the question, can the training and deployment of the tool play a role in how well we can do? And this point will come up in some of these examples. This is, when you're using an AI-based intervention, it's not done in isolation. It's multifaceted. It involves this human-AI interaction, and that in itself needs special study. So I'm going to illustrate this idea with another trial, which is also a multifaceted trial, looking at an AI diabetes tool to improve diabetes management. So this is a study called the 4T study, which stands for Teamwork, Technology, Targets, and Tight Control. And the idea here is that we can use data from a patient's continuous glucose monitor to prioritize who needs more attention, who needs more care. And we wanted to evaluate this multifaceted tool. Again, just like in the colonoscopy study, we wanted to evaluate it in the real-world clinical setting. And unlike the colonoscopy study, this was really exciting because we actually found that we did move the needle. So we found that with our intervention, which involved early adoption of a continuous glucose monitor, it also involved participation in a remote monitoring intervention, which meant you had to agree that we would look at the data and we would intervene if needed. We demonstrated that we could move the needle. So here I'm showing you, on the right-hand side, I'm showing you a plot of hemoglobin A1C, which is a measure for how well your diabetes is controlled. We want to keep that below 7.5% because for values above that, it sort of correlates with some worrisome clinical events. So we usually like to keep it below 7.5%. And here I'm showing you two lines. The black line is the control group. The red line is the intervention group. And you can see with the control group, quite naturally, when you come in and you're first diagnosed, your values are high. We're able to, with treatment and attention, bring that down. And you can see over time, it gets challenging to keep it in target. But the red line with our intervention shows that we were much better. Similar shape, you start out high, we bring you down, and we're able to manage it a little bit better. So this is really exciting. We have an intervention that works. So now we would love to get this adopted by other sites. But the question is this just like a Stanford thing? Does this depend on how our diabetes coaches were engaging? Does this depend on our patients? What are the drivers of success here? And again, we have to be wary of, and we have to be thoughtful about this human AI interaction. And now this is a little bit more complicated than the colonoscopy trial. We've got the patient who's giving the data into the tool is talking to the medical team's talking back to the patient. There's a lot of synergy here. So we have to ask the following questions. How early should patients adopt the monitor? How often should the medical team review the data? And how should the medical team deliver messages to patients? You know, we had someone on our team who has diabetes, so she has the lived experience, and she told us, you know, if you deliver these messages so frequently and in this manner, it can be really demoralizing. So we have to think about how these messages land with the patients. So we can use tools like micro-randomization in our design to study all these multiple components, and I'll give you an example. So here's one way that we can think about how to design a study to look at all of these different components that go into the intervention. So we can take someone who's diagnosed with type 1 diabetes, we can randomize them to one of two groups where you adopt the monitor immediately or you adopt it sufficiently early. And we can evaluate whether adopting it sufficiently early is just as fine as adopting immediately. We can play the same game and randomize those patients to getting a weekly review of their data or a monthly review. And hopefully the monthly is just as fine as the weekly. And then here's where the micro-randomization can come in. We can look at your current glucose values. And we can say, okay, if we assigned you to monthly, and you're not doing okay, you're not managing your disease okay, can we randomize you to meter up that frequency or not? And we can look within that subgroup whether or not a tailored intervention could work. And we can play the same game among the weekly. If you're doing just fine, maybe we can meter down the review. And so this is the way that micro-randomization can allow for those questions to be answered within subgroups. And I realize I've been talking about this human-AI interaction where the humans are sort of patients or the medical team. What about when they are the data scientist or the clinical trialist? So that's what I want to talk about in this next example here. So in this next example, this is a breast cancer study that I'm involved in called the Terpsichore trial, where we're studying whether some subtypes with a breast cancer that have sort of a favorable form will benefit from a tailored treatment. And I know some of you are working on the Datathon. How many folks are working on the Datathon, which is focused on breast cancer? Yes. Okay, great. So you know that breast cancer it's a heterogeneous disease, and it has multiple subtypes. Well, Christina Curtis's lab has been working hard on stratifying AI into even further subtypes within even the favorable subtype of breast cancer, because even that is heterogeneous. And they're asking this question with this trial. We're asking, if we tailor the treatment to some of these subtypes, will that be beneficial to patients? And so we don't want to stop progress. I mean, the data scientists are hard at work. They're getting more and more data, and they're able to make progress on their stratifier. So what do we do as the AI stratifier for classifying patients improves over time? We want to do right by our patients, and we want to use the latest version of the AI stratifier. This might be fine if the AI stratifier improves precision, our ability to classify patients into subtypes. But we have to be really careful if the AI stratifier alters our target population, because if it does so, it can alter our ability to measure the effect of tailored treatment. And I'm going to show you an example. I'm going to illustrate this with an analogy where the patients are weather and the subtypes are rain and snow. So let's say we have an AI stratifier that is pretty good at classifying patients into subtype rain, subtype snow. Sometimes it messes up and snow gets into rain and rain gets into snow. And so the data scientists come up with another version, and they're more precise now. And so it's less likely that snow is going to get into rain and vice versa. So this is great. But what if they make such great improvements and they find, no, these are the subtypes. It's actually blue rain, purple snow, and something called green hail. And this might be true. This might be the way that we should be stratifying patients. These might be clinically meaningful different subgroups. But midway through the trial, we cannot use this, otherwise it will water down our ability to detect a treatment effect, even if there is one. So if we know that there's a treatment effect that's tailored to these subgroups, it will be very challenging for us to evaluate that. So that's something that we need to be wary of. And now I want to talk about another component that interacts with the AI tool, and this is the component of data. So we have data going into the AI tool. We have data coming out of the AI tool to potentially intervene and to evaluate the tool. And a lot of this data that we're getting to do this has been enabled through digital health technology, right? I mean, I'm sure a lot of you here are wearing your smartwatches, you've got your smartphones, you might have an accelerometer on you. This is wonderful. This allows us to enable these wonderful AI-based interventions. The issue is that there are challenges with using that real-world data. These data were not intended for research. So they tend to be noisier than data that are generated from traditional trials. And furthermore, now there are multiple data streams that need to be integrated into the study database, and that becomes a challenge. And so I'm going to illustrate ideas here with the Apple Heart study. So this is a study that I was involved in where the goal was to evaluate the ability of the irregular pulse notification algorithm to identify a serious heart condition called atrial fibrillation. And this is how it worked. So on the back of the watch, there's an optical sensor that detects waveform passively to measure heart rate. And detection of a pulse irregularity can often be consistent with what we call atrial fibrillation. And here's how it works, how the algorithm works. So at opportunistic intervals called tachograms, your heart rate gets measured. And this is like every couple of hours, unless something interesting is found, in which case it's going to increase the frequency of the measurement. If you get 5 of these in a row, you would get notified and told, go see your telehealth doctor, and we're going to give you a gold standard heart monitor to really figure out what's going on. So one of the biggest challenges in this study, so first of all, I should tell you, in this study, we enrolled over 400,000 participants. That is the largest trial I've ever been involved in, just massive amounts of data. So we enrolled using an app. And the challenge was in identifying the unique number of participants. So we were getting pieces of data that looked like it was coming from another participant that was actually mapped to someone who was already enrolled. And this was happening for a number of reasons. And I'm showing you some of the pathways here. Like, the app would update or something like this, and then you would re-enroll, and it looked like it was a new patient, or you would change your device or something like this. So we had to develop and evaluate in real time a new algorithm to deduplicate the record so that we could understand how many patients we had in the trial. I'd never had this problem before of knowing how many patients were in my clinical trial. So it raised a really interesting question, which is, are we now, if we're going to be enrolling using apps, are we now going to be estimating the sample size of our trial, like with uncertainty? Which means we'd have to really understand the properties of our algorithm to do that. And this issue was brought upon because of a profound effort to coordinate all of this data that was involved in so many streams of data for integration. So we had data coming from the phone, we had data coming from the watch, from the gold standard heart monitor, from the telehealth doctor, from surveys, so a lot of data that needed to be integrated. And what was key, so in addition to patient identifiers and device identifiers, what was really key with all of this data integration was timestamps. So we needed to understand what time the data were coming in so that we could stitch the data back together longitudinally so that we could address our study objectives and our study questions, and so that we could understand the integrity of our study, the conduct of our study, and the integrity of our data. And I'll give you one example here. So this is one of our goals was to make sure that the signal from the watch matched with the signal from the gold standard heart monitor. And you can imagine if you were misaligned just a little bit because the data were noisy, because one of the devices had a low battery life that added noise, we could be, and you misalign, you can say that the data are discordant when they're really concordant. So that was really important from an integrity standpoint. We had to know windows within which the data had integrity for us to do these analyses. And so this brings up the issue that participant behavior, device behavior has an impact on the data, right? The participant might not wear the watch. They might change the clock. The device might have an update, or it might have low battery life. All of that impacts the data that are going into this tool and then ultimately intervening and being used for evaluation. What we found was really important were pilot studies that emphasize the data integrity through joint evaluations of the longitudinal data that are used to measure treatment effect. That's really, really important. And of course, and I think many people have alluded to this, it was really important that we work with the app engineers on that. So I've given you a lot of lessons that to be learned from these trials. The biggest lesson was human AI interaction is really critical. There are so many players involved, from the clinical trialist, the patient, the data scientist, the medical team, and the data. And all of these synergies among these players really need to be considered. So for the AI-supported intervention to be successful, you have to think about the human-AI synergies when you think about the intervention. They are part of the intervention. And in order to evaluate it, you have to actually measure those synergies and study them. And I'll end with that.
Thank you so much.
Thank you.
Wonderful.